This animation illustrates that receptors are proteins that receive and bind to a specific molecule called a ligand. ARXR and PPR gamma are called nuclear receptors because they act in the nucleus. Nuclear receptors are different from many other types of cellular receptors, for example those at the cell surface in this animation, in that nuclear receptors bind directly to a piece of DNA in the nucleus. Technically, they are termed ligand-modulated transcription factors. This means when the receptor binds to its ligand, usually a hormone such as estradiol or cortisol, the receptor-ligand complex binds to DNA and turns genes on or off directly. What's key here is that the ligands that bind to these nuclear receptors are nearly all small, fat-soluble molecules that roam the body pretty freely. They get into cells without needing to be specifically transported for the most part. Most nuclear receptors respond to low levels of hormone. That is, it doesn't take much to get them to turn their target genes on or off. This means that regulating the concentrations of the hormones is very important. Although most nuclear receptors bind strongly to one or a few ligands, endocrine disrupting chemicals, EDCs, that are similar to the genuine ligand can also bind to the receptor, for example the estrogen receptors, and cause target genes to be turned on or off as you can see in this animation. EDCs that activate or inactivate ARXR and PPR gamma at the wrong time during development can disturb adipogenesis. This tells us right off the bat that it might be a little easier than we would like to disturb lipid homeostasis, leading to increased weight. Let's take the example of in vitro effects. We already know that there are chemicals that, when given to human beings, shift the body's lipid balance towards storing more fat. An excellent example is a class of drugs called thiazolidinediones, like Actos and Avandia, which are given to type 2 adult-onset diabetics. This phenomenon has been well documented for more than 15 years. The drugs activate PPR gamma, increasing the expression of its target genes. This has two effects, one intended and the other not. In some cells, activation of PPR gamma increases insulin sensitivity, which reduces blood sugar. However, the drugs have the unwanted side effect of increasing both the number of fat cells and the amount of fat stored in them. Why? It's because of what happens when those genes get turned on. If you activate ARXR PPR gamma, you turn certain susceptible cells into fat cells. In other words, the genes that get turned on make proteins that tell cells that weren't sure what they were going to be when they grew up that you are now a fat cell. PPR gamma is the master fat cell regulator, and its activation controls the body's adipocyte number, size, and function, and it increases the storage of fat into existing fat cells.